As Fred said, I'm from Cornell University. I've been working with population dynamics of trout in New York for about the last 4 years, and more recently focusing on the Adirondacks, brook trout and lake trout in particular. And today I'm going to talk about a place called Honnedaga Lake, which is not to be confused with Onondaga for Syracuse folks. And Honnedaga is actually a pretty interesting story. So it's in the southwestern Adirondacks, near the town of Old Forge. And historically, it supported a fairly diverse fishery. I mean, relatively speaking. Originally it was just brook trout, lake trout were introduced, creek chub, white sucker. But then, starting in about the 1950s, acid precipitation began to be a problem. And we saw species dropping out one by one. Until in the 1970s, it was only brook trout we were seeing, and then those disappeared from the lake proper as well. We went through a period of about 20 years where we didn't see any fish. Actually, for a bit, we even stopped surveying. But then the Clean Air Act of 1990, or Clean Air Act Amendment of 1990, came about. We started seeing pH rising, restoration in a lot of Adirondack lakes, at least chemical restoration, or recovery. And we started to get reports that people who had camps on Honnedaga were seeing fish again. And what it turned out was happening was that the population of brook trout that were originally indigenous and the only fish in the lake were coming back. We think from buffered refuges in these tiny, 1-2 foot wide tributaries. And I'll note that this is a heritage strain. If you were listening to Matt Hare's talk yesterday, Honnedaga was one of the strains in there, and it was shown as distinct from any of the stock strains or the other wild strains that he looked at. So what we're seeing here is natural recovery and essentially a second chance to go back to what the lake was prior to human colonization. And we're interested in seeing the dynamics of this recovery. How far along is it? How is that going to be happening? As well as some of the kind of spatial and size structure aspects. And it's not often we really get to see this kind of natural recovery where we didn't do anything like stocking to bring it about. So we've started a 3-year study. We're 1 year into it to really investigate this and see what's happening. And what I'm going to talk about is some wrinkles we've discovered during the first year. It's a map of the lake. I apologize that Honnedaga is awkwardly shaped for displaying on the slide. It's long, it's narrow, it's about 6 kilometers total. It's a deep lake. It's almost 200 feet deep. It's got steep sides, average depth of about 60 feet, an area of 340 hectares. So this has a lot of cold water refuge. If you're a brook trout, this is a fantastic place to be, but it's not a pool to with acid like it used to be. And I would say that there isn't much else like this in this region, where you have just a single heritage strain in a very large lake with minimal human impact. So how are we assessing this? How are we looking at this? The whale we talk about today is the trap netting that we do in spring of each year, in early May, just after ice out on a normal year. I suspect that might not be just after ice out this year. And we're using 4-occasion capture Mark-recapture, which you're probably familiar with. We go out, we catch fish, we mark them, we release them again, we come back, we look at the proportion of marked fish and unmarked fish that we get. We mark all the unmarked fish, set them out again for a total of 4 captures. And we're doing this with 14 trap nets distributed throughout the lake. 10 of them are the Oneida style nets that you may be familiar with, as well as 4 smaller hoop nets. The idea being that maybe we could get some smaller fish that we might be missing in the larger trap nets. And we're marking these fish with pit tags. Partially because we really like the internal tags. We moved to that from Floyd's and Hallprins because there's less chance for irritation once they've healed up. And because we're doing some remote sensing stuff with both large arrays and some smaller ones that I talked about last year. So there's added benefits. You may be familiar with trap nets, but if not, I'm just going to quickly walk you through it because it's pertinent to the later part of the presentation. So a trap net consists of a lead projecting perpendicularly out from shore, right about here. which forms a mesh wall that leads to a box out at the lake end that fish can enter but cannot escape from. And then there's two wings projecting out at 45-degree angles from the corners of that box. And the theory behind how this works is that in the springtime, brook trout are shallow and they're moving along the shoreline. And their natural tendency when they encounter an obstruction is to turn out towards deeper water, follow that obstruction until they can get around it. In this case, they can't get around it. They end up in a nice little box. We come along, unzip it, pull them out, zip it up again, and it's reset for another occasion. This is a great way to catch a bunch of fish with relatively low stress to the fish and low stress to the researchers, which are considered more important. So, starting out with basic capture statistics, we had a total of 399 tags out that we could recapture. By this I mean the first 3 occasions when we tagged fish. We did tag fish in the 4th occasion because we're doing a longer term study, but obviously we weren't able to see those fish again. 387 of those were tagged in the spring, and then 12 of those were actually recaptured from a fall occasion, the previous fall we were doing a pilot study. But we can't learn anything from the fall to spring period for the method we're using here because we really don't know what the survival was. We don't know what the potential population of tags out there is. We got 64 recaptures during that period, which is 16% of the tags we could recapture. And 19 of those were recaptured at the same location. So we netted them there, and they showed up again at that same net, either the next occasion or one afterwards. We also got 47 that were trapped at one location and then trapped at a different location. You'll notice these numbers don't quite add up to 64, and that's because some fish were unlucky and got captured 3 times. Sometimes at the same net, sometimes at a different net. Here's a gratuitous tagging photo of a beautiful brookie. We're using 23 millimeter half duplex tags. These are a little bit larger than some of the 12 millimeter tags people are using. But the half duplex and the larger size give us some increased read range, which for the remote sensing stuff that we're doing is really effective. And we found the healing is quite good with these tags. It also helps that fish in Honnedaga are wonderfly huge fish, and so most of the fish we capture are above the 150 millimeter limit we've set for tagging. Here is a map of the sites. It's each circle represents a site and its size relative to the total catch across the 4 occasions. So you can see we have a pretty good distribution of fish caught around the lake. A little more in this portion, but we had some down at this end. And all of the nets interacted with fish. So we started out doing a basic population estimate. And what we'd like to do is do this 3 years in a row, both to kind of increase our belief that yes, this is the population out there, but also to see if it's changing. There's some belief that we may be kind of at the inflection point of an exponential curve. There's a lot of habitat here at this lake, and we think that it can support a lot more fish than it currently does. So if it is about to take off, we'd really like to see that hope to capture that, so to speak. Now we're doing closed capture, or we started with the closed capture model. And the advantage of closed capture, meaning that we assume there's negligible mortality, there's no recruitment, no sort of emigration, that fish stay there in studyable population. The advantage is that we can get estimates with lower variation, and we don't need as much data to do so because we're not trying to estimate parameters such as survival. We can also extend a closed occasion out to something like robust design, where you have a set of closed occasions and then an open occasion. So maybe you're doing some spring survey, waiting here, and then doing another spring survey. And with that combination, you can estimate more than you could with either open or closed alone. And our capture occasions were as close as possible. When we picked up the fish and we reset the nets. This is in the spring, so it's reasonable to assume that a large amount of fish aren't dying in a week. They're not recruiting into the fishery. They're not leaving. There's one outlet, and we haven't noticed any spring movement there. With this, we got an estimate of about 2,900 fish in the lake. This is the population that were large enough to be tagged and within our surveyable area. And that was great. I was happy. We actually went to the club members and said, hey, you can catch the fish now. And they were very happy too. And all was good in the world. But Then I started going back and looking at the movement patterns. And I talked about captures of fish at multiple nets. I wanted to know where they were going. And what I found is this, and this is a very busy figure. We're going to spend some time on it. I'm going to kind of walk you through it. So here are the sites again that I talked about, sized by total capture. Each arrow represents detection of a fish at one site and then later detection at a different site, and the later site having the pointing into the arrow. I've also colored them by whether or not I classified it as a source or a sink. That is nets where we saw fish first and then they went somewhere else. Or nets where fish from multiple nets seem to come there later and then not leave again. And you can see we've got a strong source here and a very strong sink here, whereas these are more moderate. Fish are moving back and forth between them. Now this variation in site behavior could complicate modeling a little bit. Capture probability is obviously going to change depending on the site. If the nets, we assume they don't fish the same way, that may change the overall capture probability dynamics. And this source sink behavior also means that the capture probability for one net may kind of be a function of the capture probability for a different net. As more fish are moving away from that area and moving through this area, it's more linked, they're not independent. Another thing we noticed is that we seem to have two separate groups of fish. So in this portion of the lake, they're moving back and forth, they're all connected. And down here at this end, they're moving between the nets there. But we never saw any fish from this side or this side going back and forth. And that's interesting as well. You know, maybe they're two functionally separate populations, in which case we have to model them completely separately. That would once again be an easy answer, and I suspect as well, I think all of you, that they are not functionally separate populations. So there goes the easy answer. It could also be some sort of meta-population thing, in which case you have to do a more complex model, something like a multi-state model where you have two parts and then you've got a probability for transition between those. And then I've highlighted a few of the movements here. You can see fish that seem to be going across the lake. This violates what our general theories about brook trout movement. There's radio telemetry has suggested they don't like to cross deep water during this period. They're really following the shoreline. And when I was talking about how these trap nets work, that's the theory behind them, is that the fish are following the shoreline, they hit this wall, and they move out. But this suggests that either fish did cross deep water, or that they somehow went around the shoreline and evaded a whole lot of nets. If they're going to deep water, if they're spending any significant time there, there may be a population that we can't survey, and fish may be moving in and out of that, in which case that's what's known as temporary immigration, and that violates closure. So this gave us kind of some challenges as far as our simple closed model and the larger effort and the idea of doing something like robust design. Maybe we need to do something a little more interesting here. I'll start off with kind of some solutions and lessons learned. First of all, test your assumptions. It's really easy to say, okay, well, this is reasonable, this is fine, but you really need to look at your data. Now, closure tests exist, and they're great. Sometimes they tell you, no, it's not closed. But there's a lot of research out there, both in fisheries and in the wildlife field, that closure tests don't always work. That sometimes they don't really tell you anything about it. So it's never a substitute for actually sitting down and looking at your data. If you can tease stuff out, if you can say, okay, well, where are fish moving? You know, that's another tool in your box, and you shouldn't just say, I've got a fancy statistical test, okay, it's done. We can use open models instead, except some lower precision. Maybe we need to get more fish, higher recaptures. But we also have to forego more complex models like robust design that can tell you some really cool stuff. We could use something like a multi-state model, build a model where you've got some subpopulations, some movement between sites, but that requires building it. And if you've ever worked with modeling at all, it's really easy to sit down and say, okay, well, we'll build a model for this. And actually doing it and proving that it works and tells you what you think it's telling you is very complicated. So I'm going to end by really making a plug for something that I think in fisheries we need to be thinking about and talking about, and that's spatial models. So in the last decade or so in the wildlife field, there's emerged this idea of spatial capture recapture, spatial explicit capture recapture. It's got a few different names. And it's really led by Andy Royal, the guy who's doing a lot of the papers, a lot of the work with this. And the idea of spatial capture recapture is that it's not just what site it is that matters, but it's where the site is, and it's the distance between sites is actually a variable you should be considering. And I think this intuitively makes sense to us. You know, as anglers, we know that your probability of capturing a fish right here is probably pretty linked to the probability of capturing it over there. And it's not nearly as linked to the probability of capturing a fish way over there. That actually where you are matters, and things like distance, habitat, all that matters. And in the past in models, we just say, okay, well, site 1 and site 3 and maybe site 4 have different capture probabilities, but we don't talk about where those sites are. If you've done any capture of fish, you're probably taking GPS points at wherever your gill nets are, wherever you're fishing. At least I hope you are. But we throw away this information. We then build a Mark-recapture model, and we never take that into account. And I think with the computational power we have now, and with the existence of these models in the wildlife field, that's kind of stupid. There's a lot of information that we can be using that we've already collected. And we can extend this to include things like habitat. In wildlife, they talk a lot about resistive surfaces and least cost paths. And I think that there are some parallels to fisheries, where you've got maybe some bathymetric profiles between sites. Maybe you've got some habitat that fish don't want to cross or really kind of congregate at. And we can use these models to test those theories, get better ideas. And what you really get out of a spatial model is density, which is pretty cool. You can also get things like activity centers, and you still can get abundance as a derived estimate of that. So my hope is that in the next few years, we're going to be building this sort of model for the data we're collecting here, doing a hierarchical model that will tell us more, with more precision than we could before. And our remote sensing stuff that I'm not talking about today will build into that, so we can take a lot of data sources and throw that in there. But this is really kind of a call and a plea for people to think about this more, maybe talk more with wildlife folks, and get on the cutting edge here with this stuff. This is out there. I think we can use it. With that, I'd just like to thank everybody who helped out with this, especially the Kraft Lab Group, my funding, the Keycapper Adirondack Fellowship, private donors without whom none of this would have been possible, and the Adirondack League Club where this all takes place. So, thank you. I'll take any questions.
We do have time for a few questions.
Would you be incorporating the idea that you have different resistance to the way that they'll move to the system?
Sorry, I didn't quite hear that.
Would you be incorporating the way that they have like resistance to different parts of the habitat that they would move through, like Chris said another day?
So I'm excited at the idea of incorporating that. First of all, I'd like to build a model that just generally takes into account the spacing of that. But yes, that is definitely something I hope to incorporate. And if I don't do it, somebody else should.
Just wanted you to clarify what your occurrences were. So you said there was one in the preceding fall, but you're not counting that here. You're analyzing 4 occurrences?
I'm analyzing 4 occurrences, and I really have 3 recapture occurrences.
This is weekly spacing?
This is spacing of 2 to 3 nights in between. The trap net gets set out, it fishes for 2 nights. We come back at the same time of day, we open it up and pull out the fish, we immediately close the zippers, in which case the net is reset and it's fishing again. So there's probably 30 minutes, maybe 15 minutes where a net is not fishing. We go, we tag those fish, we drop them off out in the lake, in the same area kind of where they were, but not next to the net so they don't get caught. And we go back again. So we couldn't space this capture any closer than it is. The fall stuff was separate, but we got some tags from it, and for a larger, robust design, that would be fine.
In your map graphics, you showed all of the nets. I'm wondering if you saw any differences between the small nets and trout nets in terms of how they interacted with brook trout.
We did. there is definitely a difference. We hope the hoop nets would capture lots of small fish. They capture some small fish and lots of newts, which were not part of the study. But the larger Oneida-style trap nets really are the best way to capture this. So once again, if I was modeling, net type would definitely be a variable in there. I like the hoop nets. The other thing is they're shorter, so we can put them in water that drops off much more sharply. In this lake, that's a big problem. You might have noticed in the figure there's some gaps between where we have nets. And that's because it goes from 1 foot to 30 feet in the span of like this long. So you can't really hang it and try to bend down in the water. It's a tough lake.
You were talking about the place you're kind of going abnormal movements. Have you ever thought about the stresses from the tagging event affecting behavior after?
I have. There's a fair bit of evidence in pit tags that there really isn't too much stress affecting behavior, but it's certainly possible. I've thought about doing some sort of alternate tagging with like a halt ring tag and say, okay, well, we got 2 different methods. But I'm reluctant to do that because with all the remote sensing stuff I have out there, non-pit tags are, I won't say a waste of effort, but at least something there. But yeah, that's a good theory and something I'm definitely thinking about.
So what were the bounds on that estimate? And are you thinking it's biased high because of the factors that you're talking about?
I'm not sure where the bias is because with 3 or 4 different factors that could be the temporary immigration that could bias high, but maybe some populations in there. I don't really know where it's going. The bounds were actually a couple hundred fish on either side, so it's a nice tight estimate. It's kind of amazing for that sort of thing. But I'm inclined to trust the general area it's at, but otherwise, I don't really know at this point. Thank you.
Hey, Fred, can I interject quickly? Any other student judging forums? Anybody? Dennis? Okay.
Any housekeeping announcements that you make otherwise? That's my answer, but I will encourage everybody to stick around for the presentation of awards after this.
We announced a little while ago on Glimmerglass that they have sandwiches available downstairs to go get and bring up to the meeting afterwards.
Okay, so if you didn't hear that, there are sandwiches available for those of you who'd like to grab a bite to eat without leaving the hotel so that you can be fueled up for post-meeting meetings. Okay, so without any further ado, I'd like to introduce Patrick Schultz from USGS Lake Ontario Field Station, and he'll be speaking to us about scale morphometrics.
As Fred just said, my name is Patrick Schultz. I work for USGS, Lake Ontario Biology